So thanks for the organizing for putting together this nice online conference and give us the opportunity to still meet this year, even though the pandemic. I want to thank everybody for attending my presentation titled, Can we select for heat stress tolerant corals within a population without the occurrence of a bleaching event? So the work that I will be presenting here was conducted by the members and collaborators of our research group, Coral Assist, led by Dr. James Guest at Newcastle University. All the fieldwork was carried out in Palau in the Western Pacific with the support of the Palau International Coral Reef Center. I invite you to hear more about what we do by attending the next two presentations by Julia Strahl and Liam Lachs, and by looking for a poster from Elizabeth Beauchamp. I also wanted to mention that there's an offer to apply for a One Planet scholarship for conducting a PhD with our research group that closes at the beginning of January. So if you're interested, please get in contact with Dr. Guest. So as you all might be aware, coral reefs are affected by local pressures like ocean pollution and coastal development, overfishing, bycatch, and destructive fishing. And currently, the biggest threat that coral reefs are facing is climate change. Four massive bleaching events have been reported as a consequence of increases in seawater temperature during the last 30 years, affecting hundreds of reefs around the world. And according to the Intergovernmental Panel of Climate Change, unless that we limit our future warming to 1.5 degrees Celsius, there are going to be very bad consequences for coral reefs, which have already experienced an increase in 1 degree since the 70s. Even if we limit the warming to 1.5 degrees, it is highly likely that large areas of reefs are going to be experiencing regular bleaching events, threatening among 70 to 90% of the reefs by 2050. In addition, global mean surface temperature is projected to reach 1.5 degrees above the pre-industrial period between 2030 and 2050, depending on the model assumptions and climate sensitivity used. This means that coral reefs will have between 10 to 30 years to adapt to the projected temperature increase. For some coral species and reefs, this might not be enough time for natural selection to act and select for the most tolerant individuals. So how can we help reefs face climate change, considering that they might not have enough time to rapidly adapt to the predicted scenarios? In order to conserve coral reefs as we know them today, we need a portfolio of different strategies to tackle simultaneously the causes of the decline and to deal with the symptoms that the climate change produces on coral reefs. So as you may know, the main action that we need to do is reduce our greenhouse gas emissions. But together, other actions like the reinforcement of local and regional management programs of marine protected areas to maintain and reinforce connectivity between populations and develop plans for risk mitigations is an alternative. Active efforts to rehabilitate or restore degraded reefs or to prepare them to future climate change scenarios is another of the options to conserve coral reefs. And assisted evolution is a framework that can be used to prepare corals to face climate change with the main goal of producing stress-tolerant individuals that are pre-adapted to future stress scenarios and introduce them into natural populations. Currently, there are many research groups testing this approach, with some projects involving more human intervention than others. Our research group is focusing on testing if selective breeding can be used to produce corals with higher heat tolerance using colonies from one single population. Selective breeding is the process by which humans choose individuals with specific heritable phenotypic traits to breed together and produce offspring with the trait of interest. The approach we're taking is extremely conservative, as we are only working with individuals from one population, and we are not introducing any new genes into that population. We're just testing the feasibility of increasing the frequency of heat-resistant colonies by selective breeding individuals that are known to have higher heat tolerance. So during bleaching events, it has been observed that colonies show differences in their thermal tolerance, and some individuals are able to survive the stress event. In this picture, we can see two colonies of the same species, one next to each other, and exposed to the same stress conditions during the mass bleaching of 2010 in the Caribbean, where one is completely bleached and died at the end of the exposure, and the other one is healthy and survived the stress event. If coral heat tolerance is heritable, which is something that we are currently investigating, then gametes from colonies with high heat tolerance can be used to produce offspring to restore degraded populations or to increase their frequency in areas that are predicted to be subjected to heat waves. If this is feasible, then we could add to the portfolio of management strategies of coral reefs the use of selective breeding. However, how do we identify thermal tolerant colonies in one population without the occurrence of a bleaching event? So in order to identify colonies with distinctive heat tolerance without a heat stress, we conducted tank experiments. For this, we took 6 fragments from 160 colonies belonging to one population and brought them to the lab, leaving the donor colony on the reef. Exposure of the fragment was done in 2 independent experiments consisting of 66 colonies in 2018 and 104 colonies in 2019. And 8 colonies, fragments from 8 colonies were present in both experiments to see the consistency of their response. Fragments were exposed to a temperature profile similar to the ones reported during bleaching events. And the experimental design consisted of control tanks that were maintained at ambient temperature around 29 degrees and heat stress tanks where the temperature was increased from 29 to 32.5 in a time period of 22 days. So during the course of the experiment, we daily assessed the health status of each fragment. As you can see from this picture, from 2 fragments of 2 different colonies and how they progressed throughout the experiment, the one in the top was bleached and died at the end of the exposure, while the one in the bottom didn't change its color or did not show any signs of stress. So health status of the colonies during the visual inspection was estimated using a score that was given to each fragment. This health score varied between 0 and 4, and 0 was used to denote healthy healthy fragments where no change in their status was observed. 1 was for partially bleached, 2 for bleach, 3 for partial mortality, and 4 for death. When starting to analyze our results, the easiest way to assign a heat tolerance category was by identifying those colonies that at the end of the experiments had all the fragments dead, and we consider them as colonies with low heat tolerance, while the colonies that had all the fragments alive at the end of the exposure were considered as individuals with high heat tolerance. However, by only using the ending point of the experiment, we're losing a lot of information of the health status changes of each colony throughout the duration of the stress. For example, if we only look at the ending point in these colonies, the first 3 colonies are going to be classified as low heat tolerant, and the last 3 ones as high heat tolerant. However, after having data from 160 colonies from one population, we realized that there were different response strategies to the stress that all the colonies can show, which opens the door to a lot of questions and can have implications for selective breeding. For instance, from this group, we might prefer using colonies as parental donors of high heat tolerance, like these 2 ones, instead of the one on top of them. So in order to analyze all the data throughout the experiment, we decided to estimate a bleaching and mortality index, or BMI, with the percentage of fragments in each health score category, which results in a continuous variable that we can then compare between colonies and throughout the different time sampling points of the experiment. In this clustering graph, you can see the bleaching and mortality index and how it varies across the time for each one of the colonies. These are the 104 colonies that were exposed during the 2019 experiment, where each row represents a colony and each column represents a time point. Blue indicates values of the index that corresponds to colonies that are healthy in comparison to the rest of the colonies for that specific time point. And the red color indicates unhealthy status. So within this data, you can see that there was quite distinctive groups in the response to heat stress. And we were able to identify the colonies that we wanted to select as relatively high heat tolerance for the selective breeding experiment and relatively low heat tolerance as donor colonies. In this graph, the mean BMI values for the 8 colonies that were exposed during both heat stress experiment is plotted. As you can see, the colonies with low BMI values during the first exposure also show lower BMI values in the second exposure, while the ones that have the higher BMI values are also the ones exposing the higher BMI values in the second exposure. A linear correlation between these 8 data points explains 42% of the variation, which is what we can attribute to the current host. The rest of the variability in the response in between years might be a consequence of differences in Symbiodinium community or in the microbiome, which is something that we are exploring further. So we have used this experimental design to assess the heat tolerance of 3 different species, Coniastrea retiformis, Echinopora lamellosa, and Acropora digitifera. And for this last one, Acropora digitifera, we have also exposed the offspring resulting from selective breeding parental colonies, as shown in this picture. So in conclusion, in large healthy Acropora population, there appears to be sufficient phenotypic variation in heat tolerance to provide material for selective breeding crosses. And around 42% of the response to temperature stress appears to be determined by intrinsic characteristic of individual colonies. So thanks for listening, and also thanks for the funding body, the European Research Council.